Hi, everyone. I'm Saad Omar. I'm the director of the Yale Institute for Global Health. For those of you who don't know me, I'm very delighted to welcome you to this virtual discussion focused on updating folks on the recent outbreak of monkeypox. Obviously, the phenomenon is recent, and these sets of outbreaks are attracting attention in countries outside where it has been endemic. Monkeypox has been an issue and has been causing outbreaks for a while in several countries. Unfortunately, these things attract attention when it spreads more widely, especially where there are sort of Western media markets. So keeping that in mind, which reminds me that when you go to the so-called source of the Nile in Uganda, there's a plaque. There's a plaque that says that it was discovered by this and this British explorer. And there's a plaque next to it that says, well, actually, we knew where it was all along. This guy showed up and introduced it to the Western world. So keeping that in mind in terms of monkeypox as well, I'm very excited to have three distinguished guests joining us today, all of whom are experts in infectious diseases and global health related topics. Before moving to questions and discussions, I will introduce each of our panelists. The first one is Dr. Anne Remoyne. She's a professor and the Gordon Levin Endowed Chair in Infectious Diseases and Public Health at the UCLA Fielding School of Public Health. Dr. Remoyne is an internationally recognized expert on emerging infections, global health and surveillance systems, and vaccination. She has been working on monkeypox before it was cool, at least in certain circles. I know that it was cool for her all along. She did her K award on that, and has been working on this infection for a while. Her work has focused on the emergence of infectious disease in populations living at the intersection of animal-human contact, but primarily in the Democratic Republic of Congo, or DRC. Then we have Nate Gruba. Dr. Nate Gruba is an associate professor of epidemiology of microbial diseases at the Yale School of Public Health. The Gruba lab uses pathogen genomics and epidemiological models to investigate the emergence, spread, and evolution of viruses such as Zika, dengue, and SARS-CoV-2. And he's done some very interesting work on the genomic epidemiology of several diseases, but more recently on SARS-CoV-2. The frames you see behind him are title of the well-deserved title pages of journals where some of his prominent papers were published, and these are sort of well-deserved recognitions of his work. Then we have Dr. Akiko Iwasaki, freshly back from Japan. She's a sterling professor in more than one ways in terms of her professional street cred. She is her street cred is very sterling in terms of her scientific contributions, but she's also the named Sterling Professor of Immunology and Molecular, Cellular, and Developmental Biology at Yale and an investigator of the Howard Hughes Medical Institute. Her research focuses on the mechanisms of immune defense against viruses at the mucosal surfaces and the development of mucosal vaccine strategies. She's the co-lead investigator of the Yale COVID-19 recovery study, which aims to determine the changes in immune response of people with long COVID after vaccination, and is the director of the newly formed Yale Center of Infection and Immunity. So the way those of you who are used to joining our conversation series, the format is similar. We will have questions and answers, and I will be moderating and I'll start with these questions, and then we'll take audience questions as we go along. So the first question, Anne, you've been working on monkeypox, as I said, for a long time. And we know that the disease has been increasing in recent years in Africa. Not only that it has been around for a while, it has been increasing. Can you talk about the history of the disease, or at least the known history of the disease, and recent trends in Africa?
Absolutely. So, Saad, thanks for organizing this. I'm so thrilled to be here with this incredible panel. Let me just tell you a little bit about monkeypox in general. So monkeypox, just as a kind of an encapsulated background about monkeypox, monkeypox was first discovered in non-human primates in a colony in the Netherlands, where they were doing polio testing. Actually, they were testing the polio vaccine. This was in 1958. And that's why it's called monkeypox, because it was first discovered in primates. Monkeypox is actually a misnomer. It's actually more of a rodent pox as we know it, although we still are trying to truly understand what the host range is. The first time that monkeypox was discovered in humans was in 1970 in a child in the Democratic Republic of Congo who was 9 months old. And this was in the context of smallpox eradication. So they were investigating any case of rash illness that could be similar to smallpox, and doing virus culture and really, trying to get down to exactly what it is. That's when they discovered that monkeypox actually could infect humans. Monkeypox, then they of course were very concerned about a pox virus that was infecting humans, and so they were going to follow it very carefully. They started doing intensive disease surveillance in DRC, and they did find that there were cases of monkeypox that were occurring, although because smallpox provides smallpox vaccine and natural infection provides excellent immunity to other pox viruses, which is really the whole idea of smallpox vaccine. Anyway, I'm sure you guys all know the history of smallpox vaccine. It's not smallpox, it's a cowpox, horsepox, something. We don't really know what vaccinia is a whole different question what vaccinia actually is, but that provides protection against smallpox. So the time that we really understood the most about monkeypox was from 1981 until 1986. So just after smallpox was eradicated, they thought, hey, we better really understand about what this pox virus that's circulating in humans does. So they did intensified disease surveillance in DRC to truly understand how it was spreading. And they did this in several health zones. So it wasn't throughout the entire country. It was just in very specific places where they knew there were cases. They found somewhere around 338 cases. They investigated them carefully. And that's really what the bulk of monkeypox epidemiology, clinical presentation, like this work is really predicated on this work from 1981 to 1986, where they did intensified disease surveillance. They were giving what's equivalent to like $90 a person for any report of monkeypox. So they were really doing a lot of case investigation. You can imagine 1980s, what $90, I mean, even today, $90 is a lot of money. So there was a lot of incentive. People really knew what to look for at that time. So there was a lot better disease surveillance ongoing. And that's how they found these cases. They did a lot of modeling as a result. They did a lot of ecological studies. And what they determined at that time was that monkeypox was, it had limited human-to-human spread, that most cases were spillover from animals. They only were able to isolate it in a single animal, I think it was a squirrel, in 1984. And by the way, it hasn't ever been isolated again, except for one other time in the early 2000s in an animal in West Africa. But what they just determined was that this really wasn't going to be something that was going to spread. And the reason, and if you look at these early papers, what they say is something to the effect of, and this is paraphrase, but because what we're going to see is increased urbanization, we're going to see improved healthcare, we're going to see, this was the early 80s, right? I mean, the early 80s, we really thought that life was going to be very, very different. There was a very rosy outlook for what was going to be happening in all of these places, and in particular in a place like DRC. So increased urbanization, increased travel, more reliance on commercial food products and ag as opposed to bushmeat. So therefore, we think that the probability of it spilling over is going to be less and less over time. But of course, a place like DRC, that's not what happened. What happened was we had a complete destruction of the health system. There was prolonged civil war. People were forced into the forests. We saw a lot more hunting of the forests. The forests became hunted out. People were eating a lot more rodents. people, were malnourished. You saw all of these comorbidities. And so that's really, we saw, in fact, so I, along with colleagues in 2010, when we started our monkeypox projects, really saw this incredible increase in cases. It was totally unexpected and really different from what they had predicted. So, that just goes to show you, you always have to be updating your priors, right? I mean, that's what we do in epidemiology, is we're always thinking about what are our assumptions that we're making and how are things changing. So we had a change in epidemiologic landscape. We had a change in ecologic landscape. We've seen population explosion over time. We've now seen cases occur in Nigeria as well. And that outbreak has continued over time. And so, really where we are today is that most of our knowledge is about this from the studies that were done in the 80s. I had a study in DRC. USAMRIID had a study in DRC. CDC had a study. These were very limited scope studies with limited funding. So we have pieces of information, but we really know about it in rural hunting villages in Central Africa. The outbreak in 2017 was really a wake-up call, I think, to everybody that monkeypox, we could be seeing an ecologic change, that the host range had changed. It wasn't recognized quickly. I always draw the example of west of Ebola. You have something kind of smoldering around, like in Guinea, nobody recognizes it. All of a sudden you see it explode. And so what we've now seen, our cases just increase over, there was already an outbreak ongoing by the time they recognized it. We've seen changes in clinical presentation. We've seen all these other things. Nigeria is an economic hub. And so as a result, we started to see exportation of cases. We had a number of cases exported that we knew about. Of course, this could have been happening for a long time, between 2018 to present. And of course, that brings us to today, which now we have a global outbreak of monkeypox.
Yeah, so you mentioned that, who would have thought that we'd be talking about pox viruses, et cetera. And it is, for this conversation series, you'd be interested to know that our last guest, which we thought was the end of our semester, of our conversation series for the semester, was Dr. Bill Foege, who devised the strategy to eradicate smallpox and co-led the program, et cetera. So I wouldn't have thought, even in January, that we'd be talking about two pox viruses back to back. One thing to remind everyone, that we are, you can post your questions in the question and answer section, and would be happy to, after a series of questions that I ask, would be happy to ask these questions on your behalf from the panel members. So Nate, sort of the next question is from you, and Anne started talking about that initially. Do you think, has something changed about the virus in the way it is spread? What can virus genomics tell us about how it is evolving and spreading?
Well, thanks, Saad, for the invitation to join this panel. I primarily joined so I can get a front row seat to hear from you all, and hopefully I have something here to contribute. There's now about 1,000 or so suspected infirm cases that in non-endemic countries. And so far with the patterns and contact history of the cases, it's supporting human-to-human transmission that Anne had mentioned that can occur. But we don't really know yet if anything is substantially changed about the virus, especially to the extent that maybe that it's respiratory borne. I don't know how much of that is actually known. And it seems like, primarily right now with a lot of the cases, it is associated with close contact. So the genomics can provide some evidence here to support some of those and allow us to further dig in. First is that most of the sequence cases belong to one major clade that is a part of this West African strain, which is thought to be less virulent. And so far, I guess, supporting that is that we haven't had very many severe cases, and I don't think any deaths yet associated, at least in the non-endemic countries, as a part of this 2022 outbreak. But some have hypothesized that there might be virus adaptations that is associated with this clade that may be aiding the outbreak. However, recently, the CDC sequenced some other viruses that are also still West African clade, but they're different lineages sort of within that, and not this main one. So to me, that suggests that there's probably not something special about that main clade that is causing the outbreak. It's the most common causing human-human transmission in the endemic regions in West and Central Africa. But as someone who really likes genomics and likes to nerd out on some of this stuff, some of the findings have been really interesting. So monkeypox virus is this big DNA virus, and it evolves pretty slowly at 1 to 2 mutations per year because it has these proofreading mechanisms a lot of the RNA viruses that I study don't have. But there's some interesting signs of accelerated evolution that has occurred. So this branch leading up to the major clade of viruses that have been sequenced has about 40 mutations that accumulated over 4 years. That's about 10 times more than what we thought. But some terrific sleuthing by colleagues have pointed out that a lot of these mutational patterns actually don't really match the normal way that viruses mutate, which is when the virus is replicating, it makes some mistakes here and there, and they happen throughout the genome, and some of them are advantageous, but most of them don't necessarily help the virus. These mutations actually look like patterns that are introduced by the host immune response, these enzymes called apobecs that are known, I think probably most known for their control of HIV infections. And so these mutations that have accumulated might actually be more of scars on the virus left by the host than true mutations or signs of adaptation. But to me, what it suggests that there's some extended period of time to human transmission that has occurred. And that's what we're seeing with these scars. So to me, this indicates that there's probably a much larger outbreak happening in West and Central Africa than what was realized. And we're seeing the extent of that now through cases popping up in other countries. I would really like to get Dr. Moine's take on that to see if she agrees with me. I see her nodding, so that's helpful. So with the genomics, there's still a ton to unpack here. We can use it to help understand if transmission in different locations, if they're connected, and to help us to develop hypotheses that we can both test with clinical studies and with experimental studies.
That's interesting. And one of the things is that there's always the cost for flying blind. And it's in our shared interest to have investments in high quality epidemiologic and lab surveillance. And the fact that these investments were not made. So as an example, where was the level of seasonal coronavirus research before the pandemic? A parallel is flu vaccine and flu virus research. We didn't invest in, and frankly, a lot of ways the variants are behaving, there's an overlap, not exactly the same thing. They're behaving in an exaggerated fashion of seasonal coronaviruses, as an example, roughly stated. So we could have learned a lot of things from that. Similarly, if we had investments in surveillance and both epidemiological and lab surveillance in terms of coronaviruses, or in terms of, monkeypox, we would have been better prepared for it for the sake of countries where it is endemic and for other countries that are facing it now. So the next question I have is from Akiko. So what do we know about the immunology of the monkeypox virus and how long smallpox vaccine protection is expected to last? What has happened to our population immunity since the smallpox vaccination campaigns ended after eradication?
Yeah, thank you, Saad, for the questions and for having me on this panel too. Like Nate, I'm sitting here in order to try to learn from the other experts about what they have to say. And I'd be happy to come back to this outbreak issue if there's time later and nerd out about that too. So yeah, getting back to Saad, your question. So pox viruses in general, infections in general, induce pretty robust and long-term immunity against disease. So smallpox vaccine, as Anne already described, is a live virus related to the smallpox called vaccinia virus that normally infects other animals like cows. So while the CDC website says that the vaccination with the vaccinia virus provides full immunity to 3 to 5 years, I believe that the data are more consistent with the immunity lasting for a lot longer, even in those who were only vaccinated once. So historically, the vaccine has been effective in preventing smallpox in like 95% of those who are vaccinated. And against the monkeypox, there's about 85% protection against the disease. In addition, because pox viruses replicate slowly, the vaccine can be are proven to be substantially able to lessen the disease when given within a few days of exposure. And the correlative protection is well understood also for the smallpox, at least. The neutralizing antibody titers above 1 in 32 are associated with protective immunity against the smallpox disease. And T cells also contribute, but antibodies are sufficient to confer protection. So the routine smallpox vaccines among the American public stopped in 1972 after the disease was eradicated in the US. In Japan, where I grew up, the public vaccination against smallpox also ended in 1976. And consequently, I only received 1 dose of the vaccine. But in a study published in 2008, the researchers found that in 97% of the participants that they've investigated, there is no decrease in vaccinia-specific antibody titers that were noted with age over a follow-up period of 88 years. That's pretty remarkable. Another study found that T cell responses after vaccinia vaccine last for 30 years. So we can expect significant level of protection against disease in those who have been previously vaccinated with the vaccinia vaccine. And hopefully the same kind of protection would apply for monkeypox.
So a related sort of question, and actually I'll take a question from the audience because it's related, sort of kind of earlier than we planned to. So this question is from Edward Nirenberg. Is there any heterologous, and this is to you, Akiko, do you think, and are you aware of any heterologous protection against monkeypox virus from molluscum contagiosum virus? Any thoughts on that? Look, early in the outbreak, we should be asking broad questions,
exactly.
because, and then sort of exploring these kinds of hypotheses. Obviously, this is not purely early in the outbreak, but in this phase of the outbreak.
Yeah, so I have no data about that, but really it depends on how related these viruses are. And Anne, if you have any insights, please jump in.
Well, I think that this is a really interesting question. I don't know about molluscum contagiosum. I don't know. And it's a good question. I literally was just writing it down saying, I think that's an interesting question. Mark Slifka might be a good person to ask about this because he's the person who in 2008, I believe it was his paper you were talking about. He did some of that early work. But, what it made me think about, and these are just thoughts that I'm having, and this is all part of the scientific process, right? This is why it's so great that you guys are getting all of us together to start talking about these things, because this is how science is done and these kind of questions come up. So when you were talking about this and thinking, okay, well, T cell responses last for about 30 years. We have long-term protection. I had a paper in PNAS in 2010 where I demonstrated this long-term rise this extensive rise in cases of monkeypox since the 1980s. And really correlated it with the cessation of vaccination. And we really had almost no cases in individuals who had been vaccinated at that point. But what it makes me think about in general is what is the threshold at which we really start to see this, and will we need, and these are just questions that come into my head. If we think that there's like a certain threshold of immunity that we need to maintain to be able to avoid this. Of course, what you really want to do is you want to be able to do that in places where monkeypox is endemic right now, because it's always better to control it at the source. And this goes back to what Saad was saying at the very beginning, which is, we pay the price every single time that we don't invest in disease surveillance, lab surveillance, and this kind of infrastructure. And as my late father-in-law always used to say, it's much easier to stay out of trouble than it is to have to get out of trouble. And I really think that that's, it just couldn't be more true than in public health. I mean, that's really the point. But I just start to wonder, well, what is that threshold? And maybe we'll have to, reach that threshold again, either by natural or preferably vaccine-induced immunity by revaccinating a certain groups to be able to really achieve that threshold again. In particular, if we start to see this virus establish itself in as something that is spreading even at a low level amongst humans, or my big concern is if we see a spillback or reverse zoonosis, then we're really going to have to consider what does it take to be able to keep pox viruses at bay. And this then goes back to what Nathan was talking about, which is, what do we think about these genetic changes, and what does this mean? Pox viruses are stable viruses. We don't see a lot of, we really don't see the kind of potential for adaptation and fitness that we do, for example, in a coronavirus. That said, we do see changes. Do we know what they mean? What will they mean? These are things we need to get in front of as opposed to chasing behind them.
Yeah, no, so I was born outside the US, so I have a smallpox scar because, in a lot of parts of the world, that vaccination was stopped a bit later. But we should remember, look, these countries where even, vaccination was stopped later have really, really young populations. And so according to some estimates, even if under the best case scenario, the population immunity provided by a smallpox vaccine is around a quarter of the population because of just the population pyramid, and even and the fact that with smallpox, not only 85% protection is high. So even if that kind of a scenario, so we should keep in mind it's not just the immunity part, but also the demography in some of those countries. So coming back to Nate, what can we do? Actually, you know what? I'll start with you again, Anne. How should we respond to the monkeypox outbreak or these sets of outbreaks, including what can we learn from our African colleagues who have been dealing this for many years? And by the way, so we extended an invitation, and there was some initial interest from our colleagues from Africa CDC, but for various reasons, logistical reasons, they couldn't join us for this panel. But what can we learn from the previous experience in terms of addressing this infection and these outbreaks, and apply that to this series of outbreaks? So I use the term outbreak and outbreaks interchangeably. I do think it's a set of outbreaks that are happening that may have overlapping factors that has made this moment where this is happening. But I think we may find out that these are independent but concurrent outbreaks.
Well, I think that what you're saying is right. I mean, we're seeing a series of global outbreaks, and I think this goes back to that we're clearly seeing rises in cases in Africa in the traditionally endemic zone. And it's likely seeded outbreaks that we just haven't been paying attention to. And in particular, because the West African clade tends to have a more mild presentation, all things being relative, and could be mistaken for other things. I think that it's one of these things, if you're, staring into the dark, you might not see anything. You got to turn the light, shine a flashlight, you're going to catch something. But if you turn all the lights on, you're going to see a lot more. And I really think that this is about situational awareness. I believe that that some of these cases that we've seen in the US are actually more closely related genetically to the outbreak the case in Texas, right? So that suggests that there have probably been multiple introductions, and now that we're looking for it, we're actually seeing it. And this could have been something that's been spreading for a while, even at a very low level, but then finally reached a network where it could propagate very quickly. And now we're seeing all these cases. So your question, I think, really is so what now? What do we do now? Here we are, what do we do? And I think that there are a number of things we need to do. I mean, first of all, we need to be able to have this kind of situational awareness. So what do? You need good syndromic surveillance. You need good clinical pictures and information so that clinicians can actually make these diagnoses, so that people can identify themselves as at particular risk. You need good risk communication, so that everybody understands what the risks are, how this is all potentially going to impact them. Of course, beyond the clinical part, you need diagnostic capacity. And right now we're very hampered by this diagnostic capacity. I just saw a note that there are several cases, I think 10 cases of orthopox positive cases in New York right now. So now they're going to have to send them on for confirmation for monkeypox. And so this is a process. Listen, at least we have orthopox assays that are out there. And so we don't, that in and of itself, I think is going to be relevant. Then you can send it on for further, but we really are going to have to expand our diagnostic capacity at a local level, at a national level. We're going to have to make this something that's very accessible if we really want to be able to capture the cases. And this is just standard public health. You're going to need, ideally, you'd want to have things like lateral flow assays and rapid or point of care diagnostics. But even in the interim, really rapidly scaling this up will be important. Syndromic surveillance, communication to everybody, and then we have to have a really clear plan about prioritizing vaccines and therapeutics, and who's going to need them, how they work for people, what the plan is going to be to do this. And that really means that we need to be scaling that up as well. Now, at the same time, we have to really be scaling up all of our research, to be able to truly understand what's going on. We have to be able to characterize transmission mechanisms and its potential. We need to characterize clinical presentation and disease severity in various risk groups. We need to know what the virus does in immunocompromised individuals. We need to know, is there an asymptomatic form of infection? Do asymptomatic people transmit? I mean, we've done this dance before. We know all the things that we don't know. And I think the most important thing is to be humble about what we know and what we don't know. Because what we know is really about how this virus has been spreading traditionally in a very different epidemiologic and ecologic landscape. So I think that the key is, saying, okay, well, here's what we know so far. Now we need to go further so that we truly do understand this, so we can get in front of it, because the stakes are frankly very high. If this virus becomes, as I said before, if it becomes established even at a very low level in human populations, or we see reverse spillover, we're really going to have to reconsider how we view a circulating orthopox virus in humans. And do we think that this is enough of a risk that we're going to have to really reconsider what vaccination means? And that was one of the big benefits of smallpox eradication program, right? You eradicate a virus, you're able to prevent the human suffering, which is the key issue, but then you're also able to dedicate those resources for manufacture of vaccines and distribution of vaccines to other virus or other pathogens, to other issues. And so, it's an enormous step backwards for public health to have to really reconsider utilization of smallpox vaccine again, in public health. So the stakes are quite high, I think. You're muted.
Yeah, so you're absolutely right. And that brings me to Nate. What can we do to prepare for a local cluster if it happens? What research questions do you plan to address? I know in an ideal world, you would still be, we wouldn't have had a pandemic, we wouldn't have had a monkeypox outbreak, and you'd be working on dengue. But we all know that this will require a scientific response. And so what do you think? What should we do with the local cluster, and what questions we should ask? Especially what you think, what you are planning to ask.
Yeah. So first, I want to start by just reiterating some points from Anne that I think are incredibly important here, is that we first need a better handle on the extent of local transmission if it's happening, right? Like in our communities, it really feels like we're getting like the tip of the iceberg here. And I'm not saying it's a COVID situation where there's like, going to have hundreds of thousands of people being infected in just a short amount of time, but it's likely a lot more than what we know. And this absolutely needs to include the rollout of frontline diagnostics. So currently the testing is still encompass in the CDC's laboratory response network. So this includes more than 60 labs across all 50 US states, and most of these are located in state facilities. So if you were to present to the ED or clinician with monkeypox-like symptoms, what they would do is they would take a swab of the pox or lesion, and they would first send it to the state labs for this broad family level test. And then if it's positive there, then the samples are sent to the CDC for confirmation. They do a genotyping assay and determine if it's the West African strain as they have been doing, and then they do sequencing. And this is the similar sort of setup that we had at the beginning of SARS-CoV-2 in early 2020. This system works great for rare pathogens because hospitals and diagnostic labs cannot reasonably be expected to have tests for every single pathogen that is out there. Right? This is why the laboratory response network exists, so that when we have something strange that comes up, we can figure out what it is, and the CDC can respond around that. However, given that monkeypox is here, like we know that it's here, there are cases that are rising. They're rising around the country or around the world. It's my opinion that we really need to roll out these frontline tests and to be able to reduce both the resulting times of having tests to go from sent to a lab and then sent to another lab, and also to increase testing capacity. But maybe even more importantly, it might make ordering these tests easier, especially in the circumstances when the symptoms resemble so many other common diseases. We're hearing of a lot of cases in MSM communities where the symptoms look like other STIs. There might be just one or two pox that are located on or near the genitals. And if someone is not astute, one, it could be missed, but also just could think it might be something else. And we almost need to include that right now in general STI testing. And that can't necessarily happen, I think, in the current system that we have. But it's not really clear to me what the frontline labs would actually use, because I don't know of any commercially available monkeypox test that is approved by the FDA that we could just put in all these labs. That is a problem. So my group's first goal, just kind of how we did, I feel like deja vu a bit. I know this is not COVID, but there are some things that are related to this. And at the beginning of the COVID pandemic, our initial response was like, how do we help diagnostics in any way possible? So what we are doing is that we are just working with several different PCR tests to optimize the different conditions, to do comparisons, to allow different diagnostic labs to be able to look at results, select what tests they want to do for internal validation, until we actually have some commercial options, available. Relatedly, we're also working to streamline the sequencing assays. Essentially, our goal is to take COVID-seq, right? All this 11 million sequences that have been generated for SARS-CoV-2 sequencing, and adapt monkeypox virus sequencing to fit in those systems, because we know labs know how to do it. There's a lot of capacity that's built up, and we need to be able to validate that so we can have, along with the diagnostics, we can have the sequencing, because we know how important the sequencing is now, I think. And then finally, if we have a local cluster, right, it's our plan to conduct clinical studies to determine first, like when and where do you find the virus in various parts of the body, different tissues, sample types and everything. And both this could help improve diagnostics, right? If we could, if for example, if there's asymptomatic transmission that is happening, if the primary form of being able to test for monkeypox virus is via a lesion or a pox on the skin, and if you don't have one, how do we test for it, right? If we can detect this in oral fluids or saliva, and if that could be a great test, that would be fantastic. We would like to be able to know that so we could roll something like that out. And also, I think that this would help shed light on transmission. If we don't necessarily know how it's happening, if there are cases where transmission is happening where it's not very intimate, close, skin to skin contact, right? We need to be able to use that so we can implement the public health practices that we know that can then stop transmission.
Yeah. So the only way I know of controlling an outbreak is to get ahead of it. And we are still following this series of outbreaks. As long as you're behind an outbreak, you have little chance of controlling it. But again, just to caution everyone that this is now, we're not looking at the start of another COVID-19 pandemic, but things can be significantly impactful even without getting to that level. I hope that doesn't become our only benchmark of paying attention to these kinds of diseases. Akiko, obviously, like any disaster movie included in an outbreak, vaccines are the heroes. And the narrative arc ends with an effective vaccine. We know in real life, the story is a little bit more complicated. What do you think about the pipeline for monkeypox, the vaccine pipeline for monkeypox? And any thoughts on that?
Yeah, absolutely. So I'm glad that the immunology is the hero in most of these situations.
Well, I would say more broadly, vaccinology. As a vaccinologist, I think immunology is part of that family of disciplines.
Absolutely. So yeah, the good news is that there are vaccines that are effective, as I already discussed, against the monkeypox. And there's one that is currently FDA approved called Jynneos. It's also known as Imvanex, which is a replication defective vaccine that's given subcutaneously. And it's a live vaccine produced from the strain called modified Vaccinia ankara. And it's like I said, it's an attenuated and non-replicating virus vaccine that can be given to people. And that is already FDA approved to prevent monkeypox disease. But the Strategic National Stockpile has other smallpox vaccines that could also be deployed if needed, such as the more traditional vaccinia virus vaccines called ACAM 2000. It's also known as Imvamune, and it's a live vaccinia vaccine that's given percutaneously. That's sort of the closely related to the original vaccine that was used in the 70s. But this one is produced in the laboratory setting in tissue culture, purified vaccinia virus that's been given to people for prevention of smallpox disease, and I think could be applicable to monkeypox disease as well. And there's one more vaccine that is in the stockpile, which is the Aventis Pasteur smallpox vaccine, which is still an investigational vaccine, but that can be used under emergency use authorization. So there are these vaccines that are known to work against smallpox that could be used for monkeypox. And as I said, there's only already one that's FDA approved for the monkeypox. And then I know that other companies like Moderna are already working on developing a monkeypox vaccine. So hopefully there'll be plenty of different vaccines in the pipeline that can combat the disease if needed.
So having been involved in the early 2000s in some sort of planning around a possible smallpox bioterrorist attack, there was a lot of discussion of that, after 9/11. And I had the privilege of being involved with some vaccine policy related, especially safety. One of our initial thinking was that people like me who have smallpox scars could be on the front line of the response because of that kind of immunity. And here, obviously, cross-protection against monkeypox, as you alluded to, that we have evidence of that, but we need to do updated studies. Do you think that could be a part of strategy? So people, one part of diversity in this country is that there are people who are older who have had smallpox vaccine, and people who are relatively younger, because they are immigrants, have received a smallpox vaccine. And this is true for the rest of the country as well. Do you think there is a value in doing those kinds of studies of the residual or ongoing cross-protection to make sure that we have a sort of a more comprehensive public health response where we leverage that diversity?
Yeah, I think that is really an important issue, especially for those who may be coming in close contact with patients with the monkeypox infection. And even though I said that, pox viruses provide robust lifelong immunity, this is just an assumption against the monkeypox right now.
Yeah.
So we do need to do much better sort of serosurveillance and, if possible, T-cell assays to see how long does the immunity last against the monkeypox, and whether we need a booster. And of course, a booster is always a great thing to do if there's sufficient vaccine available. But as Anne alluded to, the primary, like, the most urgent target population may be the people who are living in the endemic area. So again, how do we distribute the vaccine dose in a proper and equitable manner to control this pandemic? That's sort of, an important question going forward.
Right, I'm going to sort of frame a question from Elizabeth Tuckwell, who has posed that question. I think the intention is to clarify something. I know the answer to that, but I think we should address that question head on early on, which is there any lab that might have enhanced monkeypox, such as some speculated for COVID? I think we should address that head on so that there isn't an information gap where a lot of conspiracies then evolve. Nate, I'll put you on the spot for that.
So I don't think there's any evidence for that. I mean, and there's really, as Anne had mentioned too, there's not really a lot of labs that are even working on monkeypox for this to happen. But there's nothing that concerning that any sort of red flags that I have seen that would take me down this route. But of course, there's and also specifically here with the viruses from this outbreak, we have a recent common ancestor from the outbreak in Nigeria in 2017. And then the evolutionary patterns that we've seen since then make sense biologically with human-to-human transmission. So if there was any engineering, it wouldn't even be clear what that would be that would have aided anything here. So I think that would be a very remote possibility.
Yeah, and then the other thing is that just the probability of the fact, because there is a natural, the natural transmission is happening for a while in a pretty substantial endemic area. So again, if you look at that kind of a counterfactual, it is more likely, much more likely, for it to have come from a quote unquote natural source at this point.
I want to just
go ahead, please elaborate
a little bit, because I mean, listen, we've all been, if you look back at even the papers that we wrote back in 2010, not just me, our colleagues at the CDC, all of us were saying, listen, you've got to be able to control monkeypox now and really understand it, because if we see cases continue to rise, as we predict they will, we're eventually going to see importations. And that's just, this isn't a mysterious virus that we hadn't identified before. This is a known pathogen, that did exactly what we predicted it could potentially do, which is if it happened to get itself into an appropriate network, an advantageous network, that it would be able to spread rapidly. And that's what we're seeing. Now, of course, we have all of these concerns. Okay, well, what happens if it started getting into immunocompromised populations? Could we see it mutate more? If we see, this kind of, ability to continue to spread, we're gonna get given this virus some runway that it hasn't had before. But this isn't a novel pathogen that we've never seen before, and it's doing exactly what we expect it would do, having gotten into the networks that it's gotten into. The clinical presentation is a little bit different than we had been paying attention to, but it turns out there have been records of this in Nigeria, reports of this in Nigeria, that just nobody had the funds or the ability to do the kind of research to be able to document this where it was already happening. Now we're seeing it happen, on a much larger global scale. And I'm sure our African colleagues would say, hey, we've been seeing this for a while. If you'd invested in our networks and our ability to control this in a place where it is endemic, we'd already know about this, and we'd already know what the risks are, and we just haven't done that. We've ignored that possibility. And of course, this is a situation where we've come into this. I keep likening this as the same as always. My analogy for this is like we've tuned into a new TV series and we don't know what episode we've landed on. Was it 2? Is it 5? Is it 10? Are we in season 3? Where are we? I don't know. But we definitely missed some of the origin story here. But the early origin story we've got, we know where monkeypox came from. We know that this was always a possibility. It was just low on the list of priorities of something to be able to pay attention to. And even we knew there was this existential threat that this could possibly happen. I mean, it's always like the squeaky wheel gets the grease, and monkeypox was not squeaky enough to get that kind of grease. And now we are here in a situation where we've given this virus an opportunity to spread. It spreads well with close contact and fomites and respiratory droplets. At the minimum, those are the things we know. And hey, like, what could be better than sexual networks for that? It just makes sense. I think at the end of the day. So
I know. So again, actually, I'll take the liberty to put you on the spot. And in terms of, because we have a lot of people who are early stage investigators and students who attend these events. So tell me a little bit about your experience with getting resources for monkeypox, and how, and your case study, to my mind, is a microcosm of how governments and international entities should invest better in this kind of work. Because your K award, which is a primary investigator, early stage investigator award, was on monkeypox. So would you care to elaborate a little bit on what you had to face?
Sure. So, I came to this from our common ancestor, our common advisor, Mark Steinhoff at Hopkins. And I went to NIH, actually. So I was at the center at NICHD, and I was a program officer on a project in DRC. And when I was there, I recognized that there were cases of monkeypox there. And because I'd worked in disease eradication as a Peace Corps volunteer, and then I worked in polio eradication, I worked with Mark on pediatric infectious diseases, vaccine preventable diseases. I was really attuned to this issue of vaccine preventable diseases. And when I met with my now very long-term collaborators and close friends, Jean-Jacques Moyembe, and also the late Emile Okito-Landa, the Dean of the Kinshasa School of Public Health, Director of National Lab, Moyembe, they mentioned that there were all these cases of monkeypox that weren't being reported. And I thought that this was really important and interesting. But at the time, there really wasn't a mechanism to be able to send them to get tested. And the Congo didn't have that ability. So we sent them to the, just using, we scrounged around for some funds, and were able to send them to Germany for the pox coordinating lab that was able to do these analyses. And we were able, it was our first paper that was an EID, just really demonstrating that we were seeing increasing cases of monkeypox. So we were able then to leverage that to get a grant. It was, at the time, it was a sizable grant to be able to look at the increase in cases of monkeypox. And so we set up enhanced disease surveillance in some of the early sites. I mean, it wasn't perfect. We did the best we could. We did some serosurveys. And we really found this increasing rise in cases. Meanwhile, I got my K on monkeypox, epidemiology of monkeypox. And when I finished, in order to be able to continue this work, I was going to need more than $500,000 for 5 years to do this kind of work, to be able to do the epidemiology work, to be able to support the infrastructure in DRC, to be able to do that kind of intensified disease surveillance, to be able to do the kind of molecular epidemiology that needed to be done to be able to have that lab capacity. And that's like far beyond that. And so I really, wasn't going to be able to sustain that as a junior investigator, writing my first R01 afterwards on that. And because I didn't have all of this infrastructure that I was plugging into, this was really infrastructure that I developed on my own. It just seemed overwhelming and impossible to do. And so, we kind of pivoted to other things for a while, and we'd always been working for a low level on monkeypox, working on the data, publishing our results. We had little projects here and there to be able to continue to do active surveillance for several years. And it was only a couple of years ago that we were able to get funding to do, real to be able to get back on the ground and really do a more robust project on monkeypox again, just, over the last couple of years. And that was because there was an outbreak in Nigeria, right? So all of a sudden there was some renewed interest that we saw this shift, this, explosion of cases there. And so all of a sudden it was clear that this was something that needed to be paid attention to at some level. But, it's really hard to do this kind of work in low resource settings and to be able to do the kind of enhanced disease surveillance that needs to happen. I mean, governments can't pay for that. I've heard from colleagues in Nigeria, there are many quotes of them saying, listen, we knew that there were a lot of cases out here. We couldn't do anything about it. In DRC, it's the same thing. I mean, they don't have the resources. What they're able to do is case counting, but they can't look at transmission. They can't do case investigations. They can't do all the kind of things that need to be done. I mean, it's very expensive, very consuming work. And, DRC has been very consumed with a lot of other very serious outbreaks to contend with, including, back-to-back Ebola outbreaks and other things that have really, I really have to give credit to my Congolese colleagues who are just heroes, who have worked themselves to the bone. I mean, I see this burnout and how tired people are from COVID. Well, just imagine with these incredible people working, with many fewer doctors, many fewer researchers, many fewer resources, just doing incredible work on the ground with limited resources and combating multiple outbreaks all at the same time with very little funding and very little resource. So, I mean, I think that this just goes back to, you pay now or you pay later. And you got to pick what price you're willing to pay and when you're going to pay it. And we've often said, oh, well, we'll pay on the back end as opposed to paying up front. And, that costs us dearly in so many ways.
So one last question, we are sort of, before I wrap up, and a quick question, but there's, several people are asking the same question in different ways in our question and answer box, which is, and Akiko, I'll ask you this question: should the general public get vaccinated? Again, with a smallpox vaccine, presumably, or a smallpox, an MVA vaccine tested against monkeypox. But generally speaking, should the general public think about getting vaccinated?
Well, at this point in the pandemic, I don't think that's quite necessary.
Don't use the P word, Akiko. I know that it was.
I'm sorry. At this point, I'm sorry, not pandemic. At this point of the infection that's going on, I don't think the general public needs to be vaccinated. Of course, we can never underestimate the viruses, right? There may be future variants that, even though it's unlikely, could be more transmissible or whatever, and at which point we do need to reconsider these kinds of equations. But I think it's not at the point where we have to worry about the general spreading of the virus everywhere. But we do need to really keep an eye and potentially vaccinate the people around the transmission sites and bring vaccination and other strategy that make more sense to contain the spread of the virus throughout the world. I think that's a better approach than doing general vaccination.
So a couple of things as I wrap up. So I do believe we should build the button but not press it. I do believe that we need to make investments and prepare, even with large groups, not falling, even if you fall far short of a general everyone getting vaccinated program. And if we have a large set of high-risk groups, etc., even that is a substantial investment. So we should do that absolutely now. But also we should prepare for a few other things. First of all, because it is the initial clusters are identified in MSM communities, we need to get ahead of any potential stigma. We can't, just repeat disease after disease where based on ethnicity or sexual preference, because remember, a lot of these things are identified in certain sexual networks, but it's not the virus, it's the initial identification that is happening. So we don't know. So there may be certain risk factors associated with different kind of contact, but we need to address stigma because it's in all our interest that we get ahead of this virus through collaboration and cooperation of various communities. And it is a moral responsibility to address it where it is impacting. The other thing is we need to get ahead of misinformation. And we need to do that. And we need to have a robust research agenda. And our group received the first IRB approval of our first study on this series of outbreaks 2 days ago. And so we're already in the field, etc. Not in the field in DRC, etc., but it's a different kind of study. So we'll have to have this robust agenda. With that, I'll wrap up. with only 50 seconds over our time. I'll wrap up. Stay tuned for our fall season of the conversation series, and likely to have. And we know that the speakers we are lining up will attract a lot of interest. So look forward to that. And just to repeat, Akiko did not mean to say pandemic at this point. But things can be highly disruptive even before, without being a full-on pandemic. With that, thank you. And thanks for joining us. And obviously, I couldn't get to all the questions, and they were really nice questions. I wish I had the time.
Thank you.
Thank you.
Thank you.